Hello, my name is Pranav Singh. I'm 16 years old and this is my entry to Youtube SpaceLab. My investigative question is, how does microgravity affect the genetic recombination rates of Drosophila Melanogaster? Thomas Morgan discovered linked genes through a study of Drosophila Melanogaster and discovered that the wild type was a gray body with normal wings and the mutant type was a black body with vestigial wings. Here are the genotypes for the mutant type and the wild type. Morgan crossed the heterozygous wild type with the homozygous mutant type, and following Mendelian genetics, we would expect to have an even 25% distribution of the expected phenotypes. However, Morgan discovered that the gray bodies and normal wings and the black bodies with vestigial wings seem to be inherited together. He noticed that only 17% of the offspring had gray bodies and vestigial wings and black bodies with normal wings, as opposed to the hypothesized 50%. He then deduced that body color and wing shape were linked and that were usually inherited together, and that those who were in the 17% category were genetic recombinants. Hypothesis: the microtubules of the cytoskeleton begin to degrade upon contact with microgravity. Kinetochore Microtubules are responsible for the segregation of chromosomes during Anaphase 1 and Anaphase 2 in meiosis. Since these might begin to degrade, I believe we will see abnormal chromosomal segregation and an increase in the hypothesized 17% genetic recombination rate. Step number 1. Obtain 40 phi's, 20 male heterozygous wild type, and 20 female homozygous mutant. Using habitat number 1 in the flute phi configuration with the following modifications: a white background and an external vacuum. Step number 3. Place the phi's in the chamber in the back of the habitat up until space. Step number 4. Once in space, release the phi's into the main habitat and allow them to mate. Once the phi's mate and lay their eggs, using an external vacuum, suck out the adult phi's, leaving the offspring in the original chamber. Allow the larvae to grow for 10 days, and then using the onboard camera equipment, monitor the phenotypic ratios of the offspring. I believe we will see an increase in the genetic recombination rate to around 30%.